just a bit of a summary. I'm going back to some stuff Alison's already discussed, and back to the Bcr Signaling Cascade. Sorry, maybe lost the top of the slide there, but you've seen a similar diagram to this, so Alison's done a better one, but this is basically summarizing the mode of action of ibrutinib. So it works down from the B cell receptor signaling pathway to target a protein called Bruton's tyrosine kinase. And this is the first in class which targets Bruton's tyrosine kinase, but there are other inhibitors which target other proteins similarly, including idelalisib, which is SMC approved, as you've heard, fostamatinib, which is an older drug, which is actually more commonly used in rheumatoid arthritis now, and dasatinib, which Alison undertook her PhD in investigating the efficacy of dasatinib in treatment of CLL, and in fact has been shown to be effective mostly in combination with other drugs. So because of the need for combination therapies, and we've seen that some of the trials mainly comprise combination therapy, overcoming problems of resistance and trying to optimize, avoid side effects and optimize destruction of CLL cells, we need to find more targets, find new drugs to work alongside ibrutinib as well, and idelalisib and all of the other drugs that have been shown to be efficacious. And so the target for my project has been Mtor, which you can maybe see downstream of all of these proteins which have already been targeted. Now, the interesting thing about that being downstream is that all the signals that come through the B cell receptor, if somehow they bypass all of these other inhibitors, hopefully I'll be able to stop with an Mtor inhibitor, stop some of these signals. So that's the theory behind the project. But these drugs don't only act by blocking the B cell receptor signaling. And in fact, the interesting effects which were first seen in clinical trial of the sharp rise in lymphocyte count, the sharp rise in lymphocyte count, as you can see, on initiation of the drugs, and sharp reduction in lymph node bulk and spleen size on starting these drugs. That's data from ibrutinib and idelalisib from the original clinical trials. These effects are thought not to be mediated by the B cell receptor signaling pathway, but rather through another mechanism, and I'll go on to discuss that. So you've seen this already. Alison's presented this diagram. It basically just shows that, like Hugh said, the CLL cells migrate through to the bone marrow and lymph nodes because of the surface markers that they contain. These messages allow them to travel along chemical gradients to reach their target area, which is the lymph node or the bone marrow. And these places are where the cells are retained, and they hide, they avoid destruction by the immune systems, and they also they acquire changes which make them more aggressive cells, which make them avoid chemotherapy should someone receive chemotherapy. So we basically want to push cells out of these niches, these hiding places, and that could be, again, one of the mechanisms that ibrutinib is acting. But again, these things aren't fully worked out. One of the things that has been worked out is, however, that Bruton's tyrosine kinase, the protein of interest, is downstream of the B cell receptor signaling pathway, but also downstream of this chemical gradient pathway that I've just discussed. So this may be the way that it's affecting the lymph node, lymphocytes, pushing them out of the lymph nodes and in the bone marrow. But again, this has only been worked out for ibrutinib, and will need to be worked out exactly how it's controlled, and thus finding more ways that we can block it. So the Mtor pathway is my area of interest. Mtor is a protein with two complexes and control cell survival and cell cycle initiation. And my theory is that if we can block mTOR1 alone, we can see that there's a feedback pathway which allows mTOR2 to signal unregulated and is basically insufficient. So we need to block both mTOR1 and mTOR2. Now, original clinical trials for mTOR1 inhibitors, which have already been done in CLL, showed moderate results, but there was weren't fully conclusive. And the theory behind why this hasn't been optimal is because the inhibitors don't target both 1 and 2 complexes. So this is my theory, and I'm working on an inhibitor, which you can see, here, it's called AZD8055, which blocks both of these complexes. and I'm comparing it to the single mTOR inhibitor, mTOR1, and against ibrutinib as a positive control, because we know it works so well, and also to try and investigate ibrutinib further at the same time. So my project involves three arms. I'm looking at the effects functionally, so in carefully constructed assays which mimic the body conditions in the lab to try and test the impact of these drugs on cell migration. And then I'm looking at molecular techniques to try and look at the proteins and what these drugs are actually doing on a molecular level. And then a third part, which is part of the global work of the group, the CLL group, is to look at the response of mTOR inhibition in a mouse model, which we've developed. And that's what Emily, my colleague, will talk about later on. So the first assay now is it sounds scarier than it is, but it's a pseudo-emperipolysis assay. And basically involves me sitting for about 15 hours watching cells migrate. First of all, we get the samples, which you kindly donate, and I incubate them overnight in the drugs. Then I add them to the wells, and these wells contain cells which I've laid down the day before that. These cells they release chemicals which allow the cells to migrate towards them. And then I assess how many of these cells have migrated through the layer, and thus see whether they've been blocked by the drug. And to mimic the environment that these cells live in, I've added B-cell receptor stimulation as well, and I've already shown an increase in migration with the B-cell receptor stimulation, as we'd expected. However, it's not yet significant, so I need to do more of these. And over 5 hours, I've shown that there's significant reduction with the drug AZD8055. Now, that's statistically significant. We've also seen a reduction with ibrutinib, however, not significant. And rapamycin is not as good as we expected. So this is with the addition of B-cell receptor stimulation, we've seen the same effect. We've seen an increase in migration, but which is overcome by the drug, the addition of AZD8055, which is in the red, compared to the control, which is in the grey. Then these are the pictures I've taken of many hours looking down the microscope of these cells, and you can see the stromal layer. Now this slide serves to prove how difficult it is to count these cells by eye. I mean, you couldn't begin to. So I've got sophisticated machines to count each of these CLL cells after labeling them with an antibody. But by eye, you might even get an idea that there's an increased level of migration. Maybe you can't, so I've magnified it further. You can see these cells which have migrated. But again, we couldn't rely on eye because there's no way that it would be safe for certainty. So we're relying on sophisticated machinery to count for us. Maybe you could appreciate an increase with the rapamycin drug condition there. Sorry, it's just cut off. But again, basically this is to show you what I've looked at for the last year or so. Then we do the same with another form of assay, just in case this wasn't good enough. I want to make sure that we've investigated this fully. I've tried a different type of migration assay. Instead of using cells, we put the CLL cells on top of a filter, and using the chemical gradient below this filter, allow the CLL cells to migrate towards and see how many of them, we calculate how many of them migrate. And I've shown a significant increase with BCR stimulation. And again, with the drug addition, didn't see such an exciting effect. But as you can see here, we've got a definite B-cell receptor stimulation effect in each of these conditions, which is overcome by the highest concentration of the AZD8055 drug. And ibrutinib maybe not so favorable in this one as the AZD8055. And then I've analysed a subset of samples because CLL is such a varied disease, and each sample is so different. There are populations which behave more like each other than others, so I've grouped them as to the ones that respond well to B-cell receptor stimulation. You can see a significant increase in migration in these. So the exciting thing is maybe this will be more promising an area for a specific subset of patients. So all the time we're thinking about whether there be a subpopulation of patients that would benefit more than others, and trying to refine this further. This is why we need all the samples we can get, so we can tease out subpopulations and make sure that we're not missing a therapy that might be good for one subset as opposed to another. And then, sorry, it's all lost at the top. Basically then looking at surface markers that I said that cause the cells to migrate towards the chemical gradients, looking at the effect of B cell receptor stimulation on these. And we've shown a significant downregulation of the CXCR4 marker, which has been shown out in published literature, showing that all your samples behave as they do worldwide with everyone else's data as well. So that's very reassuring. And I've seen a trend to reduction in some of the other markers, CXCR4, an increase in CD62 ligand and CD44. So this is with 16 samples so far, and I'll keep going, keep analysing, and to try and find out if some of these will become more significant. Then I've used the drug inhibitors and looking at markers. I've seen a reduction in, no effect out with B cell receptor stimulation. However, I've shown an increase, or rather an overcoming of the effect of BCR stimulation in the context of the drug AZD8055 and ibrutinib, which shows that it may be acting similarly. Again, that's very promising. This new drug may also work similarly and deserves further attention. And finally, in terms of the chemical gradient analysis, I'm looking at 48-hour incubation. So I'll put cells in for 48 hours with drug and with the chemical stimulus SDF1, and looking at whether the cells survive similarly or whether there's more of a loss rate with the drug incubations. And I've shown significant reduction with each of these drugs, but a highly significant reduction with AZD8055 at the 100 nanomolar concentration. And also a significant reduction with the ibrutinib. So this is all very promising, showing that all of these drugs have shown an effect to overcome SDF1 incubation. We need further assessment of why this is happening, looking at proteins and molecular markers downstream. So this is what the next part of my project, which I'm coming on to now, and looking at proteins which may be assessed downstream of this, called RAP1, which is involved in the cell cytoskeleton formation, which causes the CLL cells to be allowed to migrate. So this protein here is of interest for me, and I'm going to be looking at staining up cells and seeing whether the localization of this protein changes, and according to using drug therapy, and with this chemical stimulation, CXCL12 as well. So in conclusion, I've shown that dual mTOR inhibition displays a greater impact than single mTOR inhibition in the functional migration assays. I've shown that cell surface CXCR4 is downregulated by BCR stimulation in the lab, and both AZD8055 and ibrutinib overcome the B cell receptor mediated effects on the surface markers. And I've shown a reduction in cell survival, CLL cell survival with SDF1 and dual mTOR inhibition. So I've got lots more work, including analysis of the cell cytoskeleton after mTOR inhibition, looking at proteins such as RAP1, which are downstream of mTOR complex. I'm going to do Western blots to show the levels of these proteins, and hopefully moving on to work it with the mouse model as well. I'd just like to thank everyone in the Paul O'Gorman Leukemia Research Centre who supported and have assisted with this work. My supervisor, Dr. Ailsa Holroyd, and Alison McCaig, have been very supportive. Mike Leach, who's been very supportive in helping us with our work, and all of my research group, the nursing staff and the healthcare assistants, and of course, thank you for giving your samples, because we couldn't do this work without you. And just show you some of the cells I'm staining up. I hope you like. It's very preliminary, but if they weren't so beautiful, they're obviously very detrimental. Okay, thank you very much.